Hello, everybody. So we're now on week 1, video 4, which is moving from neurons and how they generate resting potentials and then action potentials. And that gets the signal down the axon to the next, or to the synapse. But now we're going to talk about how then that signal is translated across the synapse, so synaptic transmission. And we're going to cover the types of neural junctions, the two main types, neurotransmitter receptor classes, common neurotransmitters and their receptors, and then events of the synapse, and finally some connectivity between neurons and summation. So to start with, there's two basic types of synapses. You have electrical synapses, which are often called gap junctions, and this is where a channel or a pore essentially allows sodium to directly cross from one cell into another. So it's a connection between the two cell membranes, and this is a really super reliable way for one neuron to always stimulate the next neuron. And so there's not as much chance for plasticity and adjustment to the signaling between two cells connected by gap junctions. But the benefits are that there's no delay, it's very fast, it's bidirectional, and it's highly reliable. So you see it a lot in reflex circuits. You also see it in the heart, incidentally. Okay, so the other kind, and the kind that we'll largely, we'll deal with both, but that we'll talk a lot about, are chemical synapses. And these are the synapses that use neurotransmitters to communicate between the two cells. So in this scenario, an action potential triggers voltage-gated calcium channels to open in the presynaptic neuron, and that's going to induce exocytosis to happen out of that cell. And we're going to go over that in a minute. But as the neurotransmitter is released, it takes about a millisecond to diffuse across the synapse. And that millisecond represents a delay that, believe it or not, can impact fitness and the ability to respond quickly to a negative stimulus. But this kind of communication is unidirectional, and that's because you have the releasing cell that has all of the packets of neurotransmitter, and there's no, I won't say there's no exocytosis, but the system is set up to only communicate from the presynaptic neuron to the postsynaptic neuron, although there can be feedback that happens between the two cells to manipulate or influence kind of sensitivity and whatnot. What else? So let's see, it's unidirectional with the exception of some feedback mechanisms that happen at some synapses. The receptors on the postsynaptic cell obviously are super important for coordinating the response in that cell. So they're going to bind the neurotransmitter, and we'll talk about two main types of those neurotransmitter receptors. And those are called ionotropic and metabotropic. And so tropic means growing. So ionotropic means growing ions. And so these are basically just receptor channels. This type of receptor, when it binds its neurotransmitter, that causes a channel to open, and you get the movement of an ion. So ionotropic. And these types of receptors mediate really fast responses, and again, cause changes in the postsynaptic membrane potential. And they can be either excitatory and depolarize, cause a depolarization event in a graded potential, or they can be inhibitory and cause a hyperpolarization. Then you have metabotropic receptors, and metabolism there refers to the fact that it involves a protein chain reaction. And so there's a lot of, there's metabolic processes, basically, proteins changing proteins. And so these receptors are called G protein linked receptors. So their morphology and their association with other proteins is diagnostic. It tells you what kind of receptor it is. These tend to be a little bit slower because there's a protein chain reaction that takes time. And so it's a little slower than the ionotropic receptor responses. Some do end up opening or closing ion channels also, but they can have a wider variety of responses. And I've listed a YouTube video there if you want to see these things in a video. So now we'll just go over some of the common neurotransmitters and their receptors. And basically for the receptors, I'm just going to give you a couple of examples. We're going to use acetylcholine as an example. So acetylcholine is a super common neurotransmitter in the central and peripheral nervous system. It's the main neurotransmitter that's used to communicate in the neuromuscular junction between the peripheral neurons and the muscle cells. And there's two types of receptors for acetylcholine. The one that's in the skeletal muscle, the neuromuscular junction, is called the nicotinic acetylcholine receptor. So you'll notice here this ACHR. I'm just gonna highlight that on the screen. So that's a really common way to notate that you're talking about a receptor. And so whenever you see that capital R on the end, you should immediately be thinking about the receptor, so on the postsynaptic membrane. And you see acetylcholine is noted there too. So acetylcholine receptor, and nicotinic is the name of the acetylcholine, this type of acetylcholine receptor, and it's named after the fact that nicotine will bind to it, which whatever. That's neither here nor there, but that's why it's named that. And it is a ionotropic channel that essentially allows for the movement of sodium and potassium. Then you have the muscarinic acetylcholine receptor. So this is a different kind of acetylcholine receptor. It's found in different places in both the peripheral and the central nervous system. And it's a G protein coupled receptor. So if we look a little more closely, this is the nicotinic acetylcholine receptor, the one that's in the neuromuscular junction. And you can see there's two representations of its structure here. You can see that there's 4 domains that cross the membrane. So when we talk about a domain, I'm just going to put yellow dots. 1, 2, 3, 4. There's 4 domains that cross through the lipid bilayer. And then as that protein kind of folds up into its 3D structure, it essentially forms a pore that allows ions to pass through. It has a binding site, importantly, for the neurotransmitter to bind, for acetylcholine to attach. And when acetylcholine attaches, it changes the conformation of the protein in a way that then allows sodium to move through it and enter the cell. So that's a ligand-gated sodium channel. And this is going to then result in an excitatory postsynaptic potential, a graded potential. The muscarinic acetylcholine receptor is named because muscarine, another drug, will bind to this receptor. So these are just named, they're 2 different receptor types, and they're just named after drugs that happen to stimulate them. So this one is metabotropic. It's G protein coupled, which means that it has 7 transmembrane regions. I don't have that drawn here. But essentially, if you imagine a lipid bilayer, the G proteins always go 1, 2, 3, 4, 5, 6, 7. And they end, so you see there's an extracellular domain that attaches to the neurotransmitter. There's an intracellular domain that attaches to the G protein and initiates the response on down the line. Let's see. So this receptor, the muscarinic acetylcholine receptor, is excitatory for the parasympathetic pathways, so the rest and digest kinds of pathways. So if you treat somebody with muscarine, then it causes an increase in digestion, it causes lower heart rate and low blood pressure, and in fact, it can be used pharmaceutically to treat high blood pressure, but only in certain cases because it has widespread effects on the other parasympathetic processes like digestion. Okay, so in any case, there's 2 examples of these different kinds of receptors, and here they are next to each other, and I basically just include this to show. You can see the red on the left is the muscarinic acetylcholine receptor. You see those 7 transmembrane domains, and it's all folded up into a 3D structure. And you can see acetylcholine can bind on the extracellular side of the membrane, And then on the intracellular side, you can see the purple G proteins coupled with or initiating downstream sequences. So this, we're not going to go into these pathways at this point, but essentially in the gray bars on the bottom, you can see that the different parts of the G protein activate different things. And so they cause a lot of changes in protein function inside the cell. And then you can see on the right-hand side is the nicotinic receptor. There's two receptors represented there, two different ones. And they have their subunits and then have a binding site for acetylcholine. And once they open, they're allowing both sodium and calcium to come into the cell. And those, the increase in sodium and potassium, sodium and calcium is what then will cause downstream effects inside the cell. So two different receptor types for the same neurotransmitter. That's common as well. And what usually you see is that those receptor types are specific to particular places, particular types of cells. And that's how you get a different response in a different cell. Okay, so other kinds of neurotransmitters, we have amines, which are derived from single amino acids, and some examples that you're, I'm sure, familiar with are dopamine, serotonin, and then the adrenergic hormones, norepinephrine and epinephrine. You've probably also heard noradrenaline and adrenaline. They're the same things. Adrenaline is the British English version, and epinephrine is the American English version. But they refer to the same hormone, same neurotransmitter, and it can be confusing, but essentially epinephrine and norepinephrine are referred to as the adrenergic hormones, neurotransmitters. I keep doing that. I have hormones on the brain because it's what I usually do. Okay, amino acids. These are two really common neurotransmitters in the central nervous system and peripheral nervous system, glutamate and GABA. Glutamate is a really common excitatory neurotransmitter in sensory systems. The receptors are usually just simple ionotropic sodium channels. GABA is the main inhibitory neurotransmitter, and that's because it binds to and opens ionotropic chloride channels. And chloride, if you remember, has an equilibrium potential that's quite negative and close to rest. And so it's going to inhibit and prevent the cell from reaching a threshold. Other types of neurotransmitters, there are peptides, purines, gases, and lipids, all that can have neurotransmitter functions. Some of these are super ephemeral, like gases, so they're present for sometimes the order of milliseconds or seconds, like nitric oxide, but can still have really potent impacts on physiology and neural function. Okay, so just a couple of quick questions. The nicotinic acetylcholine receptor is ionotropic and causes what type of potential in a neuron? Okay, hopefully you came up with graded potential there. C is the correct answer. So the nicotinic, remember, any of these neurotransmitter receptors are typically located on the postsynaptic membrane, which is on the dendrites. And so if you're in the dendrite and you're on a postsynaptic membrane, you're thinking about graded potentials. The alpha and beta adrenergic receptors, epinephrine, adrenaline, use a G protein coupled pathway. So that means that the receptors are, hopefully you came up with metabotropic. So just remember that G protein coupled pathways involve a lot of proteins, and that is a metabolic pathway, so it's metabotropic. Okay, so we've gotten down the axon to this synapse. This is a nice visualization of a synapse sliced through the axon. And so what you can see there is I guess I can point. I don't know if I have a pointer actually. Well, anyway, so this is the the nucleus of the Schwann cell, and you can see the Schwann cells wrapping around its nice insulative hug around the axon. But we're down here in the terminal, and so you can see all the little blue dots that are the vesicles filled with neurotransmitters, and those are just waiting there at the membrane, the presynaptic membrane, to be exocytosed into the synaptic cleft, which is this space here. So you have the synaptic cleft, that's what it takes a millisecond for those neurotransmitters to diffuse across and interact with the receptors on the receiving cell. In this case, a muscle. So this is a neuromuscular junction. And so those vesicles are all full of acetylcholine. And the receptor waiting down here on the postsynaptic membrane, those are all nicotinic acetylcholine receptors. But what I want to do now is just go over in a drawing the events that lead up to exocytosis of a neurotransmitter. And so here we have our presynaptic neuron, and we have voltage-gated sodium channels that are driving an action potential to move down the axon. So that's traveling down the membrane, an action potential. And once it reaches the synaptic terminal, there's voltage-gated calcium channels present. So they're just like the voltage-gated sodium channels, except for when they open, they allow calcium to come into the cell. And remember that there's all those vesicles of acetylcholine or whatever neurotransmitter you have waiting there, right? So they're all stored and they're lined up near the membrane, but they can't fuse and exocytose their contents unless calcium is present. So calcium is going to essentially induce some proteins to called snap and snare to allow the vesicle to dock with the presynaptic membrane. And when it does that, basically the membrane of the vesicle becomes a part of the membrane of the presynaptic cell, and it just opens it up and pops its contents out into the synaptic cleft. So that's calcium-induced exocytosis. I'll just write here, these are vesicles, neurotransmitter vesicles. So then you get this exocytosis of the actual neurotransmitter, and it's going to diffuse across the synaptic cleft. Which is that space in between the two cells. And then it's going to interact with receptors on the postsynaptic membrane. And that can be a number of different kinds of receptors. So if you get binding of neurotransmitter on that receptor, then oftentimes what happens is that you get a movement of sodium or calcium into the cell, and that's going to induce a whole cascade of events. That's an ionotropic receptor, right? Because it caused the immediate entry of ions. And these ionotropic receptors can cause inhibitory postsynaptic potentials or excitatory postsynaptic potentials. I probably should have written inhibitory in a different color. We'll just write over the top. I think I can do that, yeah. And that would happen if chloride were allowed to come in. Okay, so there's going to be two different receptors. I happen to write them in the same one, which is a little confusing. But essentially, if you have an ionotropic receptor that is a chloride channel, then you're going to have an inhibitory postsynaptic potential. If you have an ionotropic receptor that is a sodium channel, then you're going to have an excitatory postsynaptic potential. And that's the same if calcium is allowed to come in. Okay, so the other type of response you can have then, or the other type of receptor, is our metabotropic receptor. So again, if you get binding there, then you have a series of protein events that cause oftentimes a second messenger cascade like cyclic AMP. And this one is metabotropic. And you can have a wider variety of cellular responses in a metabotropic pathway. So these are G protein coupled. But in any case, you get then a postsynaptic cellular response. So this is presynaptic cell. Here's the postsynaptic cell. This will typically be on a dendrite, and this will typically be the axon terminal. Okay. The other thing to note is that these neurotransmitters, obviously, if they're just going to exist forever in the synaptic cleft, then you would never be able to turn off your postsynaptic cell response. And so there are a number of ways that cells get rid of neurotransmitter. There are enzymes on the postsynaptic cell that deactivate neurotransmitter. There are recycling proteins that recycle the neurotransmitter back into the presynaptic cell, which is pretty useful. There's also other cells and blood vessels nearby, and the neurotransmitter will sometimes diffuse into the blood vessel where it'll be cleared by the blood and processed by the liver, you know, somewhere down the road and recycled into its component parts. Okay, so that's the process of synaptic transmission from an action potential arriving at the cleft and causing calcium influx into the presynaptic cell, vesicle fusion, exocytosis, and then binding at the postsynaptic cell by either an ionotropic or a metabotropic receptor. So when we draw these things, we draw them very simplistically. And that's by necessity, right? It's really hard to conceptualize all of the processes that might be happening in a neuron at a given moment. But if you look at this is the cell body of a somatic motor neuron that's almost completely covered with synapses that provide inputs and points of control from other neurons in the system. And so it's important to think about how do all of these different inputs then impact the output of the cell. So that's where we start to talk about summation and connectivity. And so we'll look briefly at kind of two main patterns we see in the in the nervous system with convergence and divergence. So this is a divergent pathway where you have one presynaptic neuron at the top there that's branching out and synapsing onto multiple neurons. And then those neurons are synapsing onto other multiple neurons. And so what you get is essentially an amplification of the signal from this neuron at the top to all of these later on in the pathway. So they may or may not feel or receive this signal from the first one, depending on what else is happening in the pathways. But that neuron has the ability, the connectivity to communicate with all of those target cells. And so this is oftentimes what we'll see when you're moving from the central nervous system to the peripheral nervous system. And those kinds, and then to the motor system, the motor units. And so this kind of pathway is called an efferent pathway. It's essentially information going away from the central nervous system out to effector cells in the body. The opposite of that is convergence. And so in this case, you have a lot of different cells that are converging onto one or some cells later in the pathway. And this is really common when this is the sensory sensory systems. So it could be touch. You know, these could all be touch receptors in the skin, and they're converging and kind of distilling down their information to fewer and fewer neurons in the afferent pathway. So these words are annoying because they're so similar, but efferent, I pronounce it efferent, soft E. That's the outgoing pathway, whereas afferent with an A is the incoming pathway. So that's the pathway moving from the sensory systems to the peripheral nervous, via the peripheral nervous system to the central nervous system. So you see convergence commonly in sensory pathways, afferent sensory pathways. And that's helpful because while it's important to be able to feel touch on every part of your body, it's not probably important to have that information keep a one-to-one representation all the way up to the brain. That would take an enormous amount of neural space in the brain, and that space can be utilized much more efficiently. And so a lot of convergence happens in sensory systems. This is an example of convergence of signals from many different synapses onto a cell body, in this case, a Purkinje cell, and we're receiving synapses in the cerebellum there. So these neurons can be really highly branched, and I think I included an example like this in that first slide of showing how neurons can really vary in form and function. Okay, so this pathway with information moving from left to right likely represents which of the following? So to start, we'll just say that there's convergence there, and convergence, the first clue is that convergence is super common in sensory pathways. So the best guess there would be B. Okay, so when we have multiple neurons synapsing onto the dendrites of a single neuron, a postsynaptic neuron, then you can have each of those inputs will have a different impact on the cell's overall output and whether or not it reaches threshold. So in this example, they're showing 3 different presynaptic axons that are all stimulating the postsynaptic neuron. So all 3 of those things are causing depolarization events, excitatory postsynaptic potentials. And so those 3 things are summing and triggering an action potential in the cell down at the axon hillock, where you get your first voltage-gated sodium channels. In contrast, some of those inputs might be inhibitory. So in this case, the blue one is perhaps releasing GABA onto the postsynaptic membrane, and that's causing chloride influx, which is dampening the membrane voltage that's otherwise being depolarized from those sodium positive inputs. And so summation basically means that sometimes if you have all excitatory events, then those could sum to an action potential, trigger an action potential. But summation can also be adding a negative impact, right? So adding an inhibitory effect on the membrane potential. And so this could have the overall effect of at least making it more difficult to reach threshold and maybe preventing it entirely. Here's an example where you have one input, and of course you can have lots and lots of inputs doing this, but the point here in this example is that you have spatial summation and you have temporal summation. So in this case, it's a single axon, presynaptic axon that's excitatory, and it fired once at time A1 there, and you got a depolarization event, but it wasn't enough to reach threshold. So it came back down as the sodium pumps restored the gradients. And then you got another excitatory event, but it wasn't close enough in time to allow those 2 events to together sum to threshold. what's required for that is that they happen closer in time, like this. So if you reduce how far apart those two events were, they still allowed the same amount of sodium ions into the cell, each individually, but because they happened close in time before the membrane potential was restored back to rest, then they summed together to reach threshold. So that's an important event that's super important in a lot of the sensory systems and learning, which we'll hopefully cover down the road. So this is just a three-dimensional view of how these synapses are actually typically arranged on a dendrite. So you can see there's a lot of three-dimensional complexity to these dendrites, and some dendrites might be characterized by having a lot of inhibitory synapses. Some might be characterized by having a lot of excitatory synapses. So you can get a lot of complexity just in how the receiving cell builds itself. What receptors does it express in its membrane is going to entirely determine how it responds to a signaling cell. That's kind of cool. There's so much room for plasticity in the nervous system. Okay, and then here's an example of, or closer look at inhibition, because depending on where the inhibition happens, if it's, if the inhibitory system, well, so in this example, we'll start here, postsynaptic inhibition. So the blue negative, we'll say that's a GABAergic neuron, so it's gonna release GABA onto that postsynaptic cell. That's going to, because of summation of chloride, negatively charged chloride ions, suppressing the depolarization event that would otherwise be happening from the red input, then you might prevent an action potential from firing at all in the postsynaptic cell. And therefore, all three of those cells, target cells that are waiting for a signal from this neuron, are not going to have any signal occur. So you fully prevented a neuron from reaching any of its target cells. That's postsynaptic inhibition, essentially suppressing the graded potential, not reaching threshold. Alternatively, you can have a different kind of degree or specificity of inhibition. If instead of inhibiting at the cell body and the dendrites and causing a complete cessation of an action potential, you can inhibit at the presynaptic axon terminal. And if you do that there, then what that's doing is, again, it's the same thing. So you might have GABA release there that allows chloride into that axon terminal, and those negative chloride ions are going to keep the voltage-gated calcium channels from opening. And so that whole process we just went through with calcium has to come in order to allow the vesicles to fuse and exocytose neurotransmitter. This is going to prevent that from happening just at that synapse. And so the other two synapses that don't have those inhibitory inputs, you can see those release the neurotransmitters successfully and you get a response. So this is a finer, it's a different type of control. I won't say it's necessarily finer. That depends on what the, what the neurons are all connecting to. But it's another way that a neuron can control or change connectivity and excitement at these different synapses. Okay, so we'll end here on a question. If you want to inhibit only the connection between the signaling cell and cell B, what type of inhibition would you need to use? So we just covered that, and hopefully you came up with presynaptic inhibition, where you can control release of a single synapse rather than all of them together. Okay, so that's the end of this kind of quick review intro section of week 1. And obviously we'll be reviewing independently topics that are related to each of our units down the road. But for now, hopefully this is a good refresher, and please definitely let me know if you're having any problems or doubts or uncertainties with any of this material, because you don't want to start off feeling that way when we go into our more specific behavioral units. All right, see you next time.